I'm Adrian Garcia, and this summer I worked under Dr. Anderson and his grad student Ryan Conway, and they are in the Department of Evolution, Ecology, and Organismal Biology. And I did my research on aquatic insects in an intermittent alpine stream. So why are they important? Well, they're important because they go and they break down dead material at the bottom of bodies of water, and then they go in and, some of them eat algae, some of them are also predaceous, but then they go and get eaten by a fish, or then they go and become adults and get eaten by the spider whose web they got caught in. And so in this way, they cycle nutrients from freshwater to terrestrial ecosystems because they provide energy for both organisms in the aquatic and the terrestrial landscapes. As I mentioned, once they become adults, they do grow wings, and this provides a new means for dispersal. And dispersal is basically any method by which the organism moves from location to location. And corridors are actually the pathways they most frequently use to disperse. And this research is most prevalent now more than ever because of the ongoing drought in the Western United States. A lot of these organisms are having their habitat dry up. So they're gonna have to go and disperse and colonize new areas if they want to survive. So our first question that we proposed was, is there an upstream versus a downstream bias for dispersal? Meaning when they become adults, do they prefer to fly downstream or upstream? And we predicted that there would be a bias, and that bias would be in the upstream direction. And this is because, excuse me, this is because they would have to counteract the flow of the stream, which is constantly pushing them down in their larval stages. And if they were flying up, then they'd be able to colonize all the area at the top of the river that they've been pushed down from. Our second question was, does abundance decrease farther from the mainstream? Basically, as we leave the mainstream, are we seeing less and less insects? And we predicted that we would, and this is kind of a theoretical heat map showing of what we thought we would find. As you can see, red indicating a higher abundance closer to the stream, and as you go further away from it, it decreases. And our third question was, do insects use dry tributaries as a means for dispersal? These dry tributaries, if they don't hold water at one point of the year, they used to hold water. And so these insects used to live, well, not the current insects, but we're trying to see if they're still using these corridors as the means for dispersal, despite there not being any water left. And the way we went about testing these questions was by getting these sticky sheets and coating them in a sticky substrate. And then we placed that, that coated sheet on a wire mesh, which was put about 1 meter high. We put 15 along the mainstream, 15 on a mountain that was perpendicular to the river, and 15 in the dry streams. The ones on the mountain and the dry streams were placed at 1, 5, 25, 50, and 100 meters out. And I'll tell you what, hiking 100 meters up a mountain was not easy, but we went out, we did two trials of this, and then we came back and counted them 3 days. After 3 days, we then came back and counted them in lab. So this is a map showing of GPS coordinates of the different locations of our traps. You can see the orange represents the dry streams. The purple ones represent the perpendicular mountain, the mountain that was perpendicular to the stream, and the main river is the blue ones. And that's the flow river going downstream. So after 3 days, we did go back, count them in lab. And because we were kind of on a time crunch, we only had 10 weeks to do this. So we actually counted species as we counted each individual species based on their morphological differences. So as you can see in the top right, actually one of the species we were able to identify was Hebecephalus. It's got yellow wings, it's got a bluish body. So it looks vastly different from this black fly. And so that's how we distinguish one species from another. We gave them an arbitrary ID, and then we proceeded to just count the total number of each individuals that looked just like them. There was no distinction between aquatic and terrestrial for most. but there were some that were pretty easy to identify because they were what we would consider common aquatic taxa. And these are pretty easily identifiable just based off their features, based off the way they look as aquatic taxa. In addition to these, we did have lots of mosquitoes. As far as the common aquatic taxa, not including mosquitoes, we only caught a handful of these. And it's interesting because I had been to the site that we were at, was the UC James Reserve in the San Bernardino National Forest. I had been there about 2 weeks prior for a different project that I was helping out on. And I saw a lot of caddisfly larvae in the stream. And when we went out to set our traps, we didn't catch a lot. So it seems like we just missed them by maybe a week. And it is also the dry season. So they don't have as much of an environment to work in and thrive in, to reproduce in as they would normally. So was there an upstream bias? There was not an upstream bias. Excuse me, there was not an upstream bias. You can see the p-value is above 0.05, and D stands for downstream and U for upstream. You can see that the two are pretty similar. Obviously, there's quite a bit more outliers in the upstream, but overall, they were not significantly different. Was there an abundance difference? Excuse me. Does abundance decrease as we go further down from the mainstream? It actually does not. It seems like there's a slope, but once we put a line to it, the R-score value indicated that it wasn't enough difference to be considered significant. And finally, for the third question, insects were in fact using dry streams the greatest amount. You can see the p-value in the right corner is very, very small. So we knew there was something going on here. We ran a post hoc two-piece pairwise t-test to further analyze the differences. And this basically just compares each individual variable. So instead of taking all three and comparing them all three at the same time, you take the dry tributary, well, it takes it and it compares it to the other two individually. So you can see that the mainstream was similar to both the dry tributary and the perpendicular mountain, but the dry tributary and the perpendicular mountain were very different from each other. And overall, the dry tributary did have the highest mean for total abundance of insects caught. Interestingly enough, despite the dry stream having the highest abundance, it was the mainstream that had the highest morphological species richness. We ran the same ANOVA test and the post-analysis test, and it was actually the dry stream that was in the middle this time. So the mainstream has nothing in common with the perpendicular mountain in the sense that they're vastly different, but the dry stream was pretty similar to both the mainstream and the perpendicular mountain as far as species richness, excuse me, morphological species richness. So overall, there was no upstream versus downstream bias. When we first started off counting, I believe the first 4 sheets, it seemed like there would be something, but it would just go back and forth. And you saw the p-value indicated that there was nothing going on there. Abundance did not differ significantly across distance, which was interesting because the sheets right next to the river at 1 meter were the same as the sheets, 100 meters out. And that was something I wasn't expecting to see. So it was interesting. Insects used dry streams the most, but it only differed significantly when it came to the perpendicular mountains. It was pretty similar to the mainstream. So as far as future directions, we did go during the dry season. So there wasn't as much water for these aquatic insects to be able to thrive in. And if I had more time, I would probably try to time it better with their emergence. So again, it seems like we just missed the caddisflies coming out from their larval stages. And if I probably had more funding as well as more time, I'd probably like to look into isotope identification. So we'd introduce an isotope into the stream. And then when you go out and caught the insects, you would instantly be able to tell whether it was aquatic and if it came from your stream back in the lab. And of course, if we had more time, it'd be nice to be able to identify aquatic versus the terrestrial insects. So this would not have been possible without Dr. Purcell and the Six Legs program. Thanks so much. The USDA's NIFA, of course, the National Science Foundation, Dr. Kurt Anderson for giving me the opportunity to work in his lab, and Ryan Conway, the grad student, for guiding me throughout the entire 10 weeks. Thank you to the RISE program. Thanks to Summer Bridge, my breakout room, who's not mentioned here, but my breakout group rather, all my peer mentors. I appreciate it a lot. I feel like I learned a lot during this 10 weeks, and I just want to thank everybody for the opportunity.